Hello students, welcome to biology class. This video is in continuity with the previous video of chapter Reproductive System. The objectives of this video are structure of ova, oogenesis and hormonal control of oogenesis. First, we are discussing structure of ova. The human egg or ova is very small, only about 0.15 mm in diameter. It is non-motile and alethal, that is, almost without any yolk. The yolk is negligible because it gets implanted within about 7 to 8 days after fertilization in the uterus and draws the nourishment from the mother through the placenta. The human egg or ova at ovulation is at the secondary oocyte stage. It contains a haploid nucleus and a large amount of cytoplasm. The ova is surrounded by radiated rows of follicle cells called corona radiata, forming a dense crown. Corona radiata protects the ova till the fertilization and gets broken down only by the acrosomal enzymes released by the sperm. Inner to corona radiata is a transparent clear zone called Zona Palacida secreted by the corona cells. It undergoes significant changes and thickens after fertilization to stop the entry of other sperms. Next to Zona Palacida, on the inner side is the vitelline membrane, also called the primary membrane, and inner to it is the plasma membrane that surrounds the cytoplasm of the egg. An ovum is said to have a polarity. The region where the polar bodies are released is called the animal pole, and the region diametrically opposite is referred as vegetal pole. Here we can see the structure of human egg or ovum. Nucleus is present. The cytoplasm, here is the follicle cells, the layer of follicle cells forming a corona radiata layer, and inner to it is a jelly coat which is also known as Zona Palacida, and here is also the presence of first polar body. Next, we are discussing oogenesis. Oogenesis refers to the formation of a mature female gamete, that is, ova or egg. A couple of million gamete mother cells called oogonia are formed within each fetal ovary. No more oogonia are formed and added after birth. A baby girl is born with a set number of primary oocytes already in prophase 1 of the meiotic division in the ovary. Further development of primary oocyte remains suspended till puberty. Now discussing the various stages during oogenesis are: first one, multiplication phase, second one, growth phase, and the third one, maturation phase or meiotic phase. Now first one is multiplication phase. The primordial germinal cells divide repeatedly to form the oogonia. The oogonia multiply by the mitotic division and form the primary oocytes, which passes through the growth phase. Next, in the growth phase, the size of the primary oocyte increases. In the primary oocyte, large amount of fats and proteins becomes accumulated in the form of yolk. It is usually concentrated towards the lower portion of the egg, forming the vegetative pole. The portion of the cytoplasm containing the egg pronucleus remains often separated from the yolk and occurs toward the upper side of the egg, forming the animal pole. The cytoplasm of the oocyte becomes rich in RNA, DNA, ATP, and enzymes. Moreover, the organelles, the mitochondria, Golgi apparatus, ribosomes, etc., become concentrated in the cytoplasm of the oocyte. During the growth phase, tremendous changes also occur in the nucleus of the primary oocyte. The nucleus becomes large due to the increased amount of the nucleoplasm. Primary oocytes are diploid in nature. When the growth of the cytoplasm and nucleus of the primary oocyte is completed, it becomes ready for the maturation phase. In this image, we can notice the changes that occurs in the primordial follicle, forming a primary follicle and then forming a secondary follicle, which is formed in the maturation phase, having the different layers. The next is maturation phase. The maturation phase is accompanied by the maturation or meiotic division. The primary oocyte undergoes the first meiotic division to produce two haploid cells. Since there is an unequal division of the cytoplasm, one of the cells, the secondary oocyte, is large and receives most of the cytoplasm, and the other one is very small and is called the first polar body. In the second meiotic division, the secondary oocyte divides to form a large ooted or ovum and a small polar body. The first polar body also undergoes the division simultaneously to form two small polar bodies. As a result of all these divisions, a primary oocyte gives rise to one ova and a total of three polar bodies. In this picture, we can see the different stages of oogenesis, multiplication phase, growth phase, and maturation phase. As multiplication phase take place in ovaries of unborn female baby, so the cells of the germinal epithelium of the ovary undergoes number of mitotic division to increase in number. Now these cells are called oogonia. They contain diploid number of chromosomes. Now in the growth phase, some of the oogonia, not the all, only some of the oogonia increase in size and undergo growth phase to form primary oocyte. Primary oocyte are also diploid in nature. And in the maturation phase, the primary oocyte undergoes first meiotic division to produce two haploid cells. Since there is an unequal division of cytoplasm, one cell formed is known as secondary oocyte containing large amount of cytoplasm, and the other one is known as the first polar body, which contains less amount of cytoplasm. Now the secondary oocyte undergoes second meiotic division, resulting in the formation of large ooted or ovum, and the first polar body also undergoes the division simultaneously to form two polar bodies. Now as a result of all these divisions, the primary oocyte give rise to one ovum and total of three polar bodies. Now all these polar bodies degenerate and only the mature ovum is functional. Next topic we are discussing is development of ova in ovary. In the ovary, the primary germ cells from germinal epithelium divide mitotically to form the oogonia. These cells migrate towards the center of the ovary. Some of the cells grow large and form the primary oocyte, while the rest encircle the growing primary oocyte and are called follicle cells. A primary oocyte surrounded by a large layer of follicle cells or granulosa cells is called a primary follicle. The follicle cells are protective and nutritive in function. A large number of these follicles degenerate during the phase from birth to puberty. Therefore, at puberty, only about 60,000 to 80,000 primary follicles are left in each ovary. As we have discussed, the process of oogenesis begins in the ovaries of a female fetus before birth. A female child is born with as many as 2 million primary follicles containing primary oocytes. Most of these degenerate during childhood, and by puberty, only about 4 lakh are left in both the ovaries. Of these, only about 450 ever mature, 1 per month, and are released from the ovary during the whole reproductive life of a female. The rest undergoes degeneration and disappears due to death and disposal by phagocytes during the reproductive years of the female. Now this is referred as follicular atresia. The phagocytes help in disposal of these follicles. So only about 450 ova are produced by a human female over the entire span of the reproductive life that lasts for about 40 to 50 years of age. In this picture, we can see the development of ova in the ovary. Starting from the primordial follicle, then it is converted into the primary follicle, covering the primary ovum by a number of follicle cells. Then these are the developing follicles resulting in the formation of mature Graphene follicle. At ovulation, the secondary oocyte is released in the follicular fluid, and finally the Graphene follicle ruptures, releasing the ova. The Corpus Hemorrhagicum, that is a bleeding corpus luteum. It is a temporary structure that formed immediately after ovulation from the ovarian follicle as it collapses and is filled with the blood that quickly clots. After the Corpus Hemorrhagicum, the subsequent structure is called the Corpus Luteum. Corpus luteum is a yellow fluid-filled cavity, and the fluid is called lutein. This structure produces hormone progesterone, which helps in maintenance of pregnancy. If there is no pregnancy, the corpus luteum degenerates and a white lesion is formed known as corpus albicans. As the primary oocyte grow further, the follicle or the granulosa cells increase in number. The primary follicle surrounded by more layers of granulosa cells and a new theca and a follicular fluid accumulates in small cavities in between the cells, and this structure is called secondary follicle. The secondary follicle soon transforms into tertiary follicle. As the secondary follicle cell grows further, the small cavities merge together and one large fluid-filled cavity is formed called antrum. This mature follicle called Graphene follicle is about 1 centimeter in diameter and protrudes on the surface of the ovary. The theca layers get organized into two: the inner theca, that is known as interna, and the outer theca, that is called externa. The primary oocyte within the tertiary follicle grows and undergoes the first meiotic division, forming a large secondary oocyte and a tiny first polar body. It retains the bulk of the nutrient-rich cytoplasm of the primary oocyte. Now the tertiary follicle, which is formed, further changes into a mature follicle, or we can say Graphene follicle. At ovulation, the secondary oocyte is released in the follicular fluid, and finally the Graphene follicle rupture, releasing the ova. Now we are discussing the detailed structure of mature Graphene follicle. The primary oocyte undergoes first meiotic division to form the secondary oocyte, and the follicle or granulosa cells further increase in number. Various small cavities containing follicular fluid secreted by granulosa cells merge and fluid-filled cavity called antrum is formed. In the Graphene follicle, following structures can be seen. First one, antrum. It is the fluid-filled cavity. Liquor folliculi, a term given to the fluid present in the antrum. Cumulus oophorus. The secondary oocyte of Graphene follicle is suspended into the fluid-filled cavity called antrum by a stalk of cells called Cumulus oophorus. Cumulus oophorus is formed by the granulosa cells. The next is Zona Palacida. It is the thick area that surrounds the secondary oocyte. It is composed of glycoproteins synthesized by both follicle cells and oocyte. The fifth one, corona radiata. It refers the radiating layers of elongated granulosa cells outside Zona Palacida surrounding the secondary oocyte at the time of ovulation. After ovulation, the granulosa cells of ruptured follicles change to form another structure called Corpus Luteum. It releases the hormone progesterone. In this image, we can see the detailed structure of Graphene follicle, the theca interna and theca externa which are covering the Graphene follicle. Then there is a layer of granulosa cells. Secondary oocyte is covered by a layer of Zona Palacida layer and then a layer of corona radiata cells. There is one more picture showing the complete development of the primordial follicle to the mature Graphene follicle and then the release of ova. Next we are discussing hormonal control of oogenesis. Various hormones play an important role in the control of oogenesis in females. Hypothalamus releases the gonadotropin releasing hormones that stimulates anterior pituitary to release its hormones. Anterior pituitary produces two hormones. First one, luteinizing hormone LH and second, follicle stimulating hormone FSH. FSH acts on the developing ovarian follicles in the ovary and stimulates the maturation of primary, secondary and Graphene follicles. The follicles produce the hormone estrogen. On the other hand, the LH acts on corpus luteum. Corpus luteum secretes both estrogen and progesterone. Estrogen are a group of steroid hormones that are responsible for the development of secondary sexual characteristics in a mature woman. In an ovarian cycle, level of estrogen show regular cyclic changes. Rising level of estrogen causes the endometrium to become thicker and more richly supplied with blood vessels and glands. In this image, we can easily understand the hormonal control of oogenesis. Hypothalamus releases gonadotropin releasing hormones that stimulates anterior pituitary gland to release its two hormones, first one FSH and second one LH. FSH acts on the developing ovarian follicles in the ovary and stimulates the maturation of primary, secondary and tertiary follicles. The follicles produce the hormone estrogen, which is essential for the uterus wall repair. On the other hand, LH acts on the corpus luteum. Corpus luteum secretes both estrogen and progesterone. High level of estrogen and progesterone inhibits the release of gonadotropin releasing hormone, providing a negative feedback control of hormone. Progesterone is also a steroid and acts on the uterus. It further increases the thickening of the endometrium for the implantation of blastocyst, that is the developing embryo. High level of estrogen inhibits the release of gonadotropin releasing hormone, providing a negative feedback control of hormones. And high level of progesterone also inhibits the further release of gonadotropin releasing hormone in a negative feedback control. Thank you so much.